Okay, guys, welcome to your next to last lab. Hooray! You've got one more lab next week, and that's it. That's your last lab. Can you believe that? Yeah. That'll be endocrinology next week. And then after that, you will have your second lab exam. Okay. And then after that, you will come to lab, but instead of a lab, you'll actually take your fourth lecture exam.
That's the final.
No, not the final. Your final will be a final in lecture, but your fourth lecture exam will actually be lab. It'll be the week after your second lab exam. So, yeah, so we're basically wrapping things up more or less. Just kind of some afterthoughts, right? Okay, so as promised, I am going to do a review for your guys' lecture exam 3 today. Let's get through the lab first, and once we get a reasonable way through, I'll go ahead and do a review for that. Like I said, it's going to be 50 questions. I'm not going to have an essay component on this one. We'll see how it goes. So 50 questions, 2 points a question. Like I said, it's going to be heavy into neuro. So it's going to be about a fourth of it will be over the musculoskeletal system, and about 75% of it, or 3/4, will be over neurology. Not so heavy in the histology part of it, more heavier into the central nervous system, peripheral nervous system, spinal cord, brain areas work, but we'll talk about that when we get there. So today's lab is going to be over special senses. So there's several special senses, and we'll talk about these in even more detail next week when we do our lecture on special senses. But the ones we'll be talking about today are going to be hearing, are going to be smell, going to be taste, going to be vision. Those are going to be the big ones that we're going to talk about today. So I have some drawings up here on the board. You have probably better drawings of these in your lab manual, but just to kind of review how it works real briefly. So when we talk about your hearing, this is your basic setup. You have sound waves, okay, so waves of energy as they propagate through the air. And then those sound waves are funneled into your ear canal, okay. Your auricle, that's the outer part of your ear, kind of funnels that sound, and it makes its way through what's known as the external auditory or acoustic meatus. That's just a hole, and then it goes into the canal, and then that sound hits the tympanic membrane, and that sound kind of vibrates the tympanic membrane, and then the tympanic membrane pushes on 3 little bones inside of what's known as the middle ear. So this is your outer ear, this is your middle ear, okay, where you have the bones. Okay, so you have the malleus, okay, that's what gets hit first, and then the malleus hits the incus, and then the incus hits the stapes, a little stirrup-shaped bone, and then that stapes hits an area on this round-shaped structure that has these little loops on the top of it, and this is known as a cochlea. And the stapes hits this little area, this little structure known as the oval window, and what it does is it kind of pushes the oval window in, and then there's a little window down here called the round window, and so when the oval window gets pushed in, the round window gets pushed out, and vice versa, so you kind of have this kind of action. And then inside of the cochlea, this little curled up organ, you have fluid that gets kind of pushed around, and then as it gets pushed around, it actually hits little hairs, and then those little hairs are able to transmit that information or transduce, turn that, okay, that wave of sound, that sound wave, that mechanical, okay, stimulus into an action potential that then goes down through the cochlear nerve and into cranial nerve number 7, which is the vestibular cochlear nerve. Okay, you guys okay with that? It's a basic thing. Some other structures, you have the middle ear is actually vented, okay, through a tube called the auditory tube. Back in my day, it used to be called the Dissection tube, and I'll probably slip up quite a bit and continue to call it the Dissection tube, but it's the auditory tube. And sometimes when you get inflammation of this tube, if you get a cold or allergies, it'll get plugged up, and then what happens to your ears? They can get really plugged up, and it feels like there's something in your ear. Or if you have something known as otitis media, you ever heard of that term, otitis? Otitis just means inflammation or infection of the ear, and media is middle, so that's a middle ear infection. Sometimes with little kids that get these quite a bit, we'll actually have to puncture a little hole in the tympanic membrane to allow that middle ear to drain, and that's something called a myringotomy. Yeah?
How come the kids get ear infections so much? What is it about?
It has to do, well, it's a lot of things actually, but it has to do a lot with how their airways are shaped, and they're just more prone to that. Basically, to how those structures are shaped, and how things drain, and how a little bit of inflammation can cause a lot of plugging up to occur. And then, yeah, they tend to, as they become more adult-like, go into adolescence, they tend to not have as many ear infections. So that's generally how it works. We're going to talk about the nuance in more detail next week with your lecture, but you do have these loops that come off of the top of the cochlea, and these are known as the semicircular canals. And these little loops are filled with fluid, and as you move around, the fluid moves around in there. And are you guys familiar with a carpenter's level? You know what a carpenter's level is? A little thing has a little bubble. It's somewhat similar to that, where if you're kind of off level, the bubble goes this way. Okay, it's a very similar mechanism that's going on in here. And what these do are these semicircular canals give you your sense of balance. Okay, a big part of your balance actually comes from this system as well. So when you have inflammation or infection of your ear, or you have some sort of problem with your ear, it's not uncommon to develop something called vertigo. You can get very, very dizzy because the semicircular canals can be affected. That make sense? Okay, so that's hearing, okay, in a nutshell. We talk about taste and smell, okay, they're highly related. Okay, a big portion of your smell comes from your taste, and vice versa. And you guys know this, right? When you get a cold and your nose gets really plugged up, what happens to your taste? You kind of start losing it, right? You lose the ability to really taste anything real well. So taste and smell are very closely related. But you have, of course, your olfactory bulb. Okay, and then your olfactory bulb sits right on top of the ethmoid bone, specifically a little bone called the cribriform plate, and then the neurons penetrate that into an area, okay, inside here called the olfactory epithelium. Okay, and basically what you have is you have columnar tissue, and then embedded in those columnar cells, you have these neurons that come down, and those neurons can sample. Okay, so these are what we call chemoreceptors. These are chemical receptors. These are what we call mechanoreceptors. Okay, so you've got chemoreceptors. They sense the chemical environment, okay, of the air coming in. And you have about 40 million different olfactory receptors, and you can detect right around approximately 10,000 different smells a human being can, which is actually not very good when compared to other animals. So a significant part of our brain, or a bigger part of our brain, is actually dedicated to which sense, if you had to guess? Vision, yeah. Big part of our brain is dedicated to vision, not so much smell. Interestingly enough, though, babies have an incredible sense of smell. Your sense of smell is much better when you're newborn. And in fact, they found that newborns don't really have great vision when they're first born, but they can actually identify where their mother is in a room by just smell. just sense of smell, even though they can't see. So that tends to go away as you get a little older. When we talk about taste, okay, there are several different types of taste. Okay, you've got salty, you've got sour, you've got bitter, you've got sweet, and we've got a newer sense of taste that we've identified, something known as umami. You guys ever heard of that? Umami, it's like a savory taste. Imagine like tomato sauce, that kind of taste with like tomato sauce or marinara sauce, kind of that savory, tomatoey, soy sauce can do that as well. So it's that kind of taste. It's kind of a newer one, okay? And basically what you have these rough, rigidy patches on your tongue called the fungiform papillae. Now these actually are not, these are just kind of rough patches, and these don't do anything with taste, but what we have is in between them and embedded in the walls of them, you actually have what are called your taste buds. And when you enlarge your taste buds, basically what you have is you have a little pore, okay, and as that food comes through, pieces of that food come into that pore, and then you have about 50 to 100 what you call gustatory, and gustation just means taste. 50 to 100 of these cells kind of packaged together in kind of a semi-sphere, and they actually have these little projections, these little cilia that come out that can, not cilia, but actually fine projections of their cell wall that can sense that, okay, so these are also chemoreceptors, and then they send that information back to sensory neurons. And then this whole thing is actually surrounded by, of course, stratified squamous epithelial tissue, like you'd imagine, okay? And these gustatory cells are actually modified, they are modified squamous cells. They are not neurons, they're modified squamous cells, so they also have to have a basal cell layer to replenish them. And that's why when you burn or damage your tongue, generally within a week or two, the tongue will heal itself up real well and your taste will come back, because the actual taste receptors, as long as the basal lamina, basement membrane is intact and can continue to divide, will kind of restore themselves. Okay. Let's see here. In general, I don't know, has anyone ever seen a picture of a tongue and then like different areas of the tongue where this area is sweet and this area is sour? I want you to put a big X through that. That's complete BS. We started learning that really since the 70s. We figured out, oh wait a minute, no, you can sense sweet, sour, salty. Pretty much all areas of your tongue have the ability to do that. So that whole taste map, get rid of that. Get rid of that. But some things you do want to kind of put in the back of your mind. In general, salty foods, okay, that taste tends to be the result of sodium channels opening up. Boy, that shouldn't really be any surprise, right? Okay, so sodium channels. Sour tends to be proton channels opening up. What could cause proton channels to open up? Well, acidic, right? So foods that are acidic or substances that are acidic have lots of hydrogen ions or protons in them, tend to be more sour. Bitter tend to be your alkalotic, your alkaloids. And this is really important because our ability to discern bitter foods is very, very good. We have a very keen ability to identify things that are bitter. It's much more keen than the other taste senses. And why do you suppose that might be? What could be an evolutionary advantage to that? Yeah, a lot of poisons in plants specifically tend to be an alkaloid of some sort, tend to be some sort of alkaloid, and so they would tend to be bitter. So it would make sense that if you want to survive, okay, that you need to be able to identify that bitter taste very quickly, because that might be a substance that could kill you. So does that kind of make sense? Yeah, it makes sense. All right. And then sweet, of course, tend to be your monosaccharides, your simple carbohydrates, sugars. Okie dokie. Moving on to vision. This is probably the most complex one, and certainly a bigger part of our brain is dedicated to the vision. So what we're going to be doing today is obviously you guys have diagrams in your book. We're also going to be doing a dissection of a sheep eye. We'll be identifying some of the major structures. And I also have eye and ear models up for you guys. Take a look at the models. Make sure you can identify all the structures I have labeled here on the board are all structures that you want to be able to identify on these models for your next exam. And then I have a few other activities. There are some Snellen charts here for color blindness. I don't know if anyone has ever done that test. It's kind of cool. You can take a look through these. And then we have some visual acuity charts, and we have some other exercises as well we'll talk about. Okay, so let's talk about the eye. I'll just give you a brief overview. I'll dissect the eye for you guys, kind of show you it's done. It's actually pretty easy. And then I'll let you guys run amok, and I'll let you run amok for a little while, and as things are kind of wrapping up, I'll go ahead and do a review for your final exam. Okay, so here's your eye. Okay, if we pull the eye out of the orbit, okay, the eye is generally fairly well protected by fat within the orbit, and then it's actually anchored by 6 different muscles, and those muscles allow for lateral movement and up and down movement. And there's a little section in your lab manual where you have to identify those 6 muscles and what nerve innervates them, okay, and what they do for the eye. So if I take the eye and I do a sagittal section of the eye and I look at it, there are several kind of components and layers. The eye is generally divided into 3 chambers or cavities. Okay, the most anterior is the outer part of the eye, known as the anterior chamber. And this is what's known as the cornea. Okay, this is what, when you look into somebody's eyes, you're actually looking at their cornea. Okay, and the cornea is kind of a transparent layer, and the outer layer of the cornea is known as the conjunctiva. Sometimes when you get an infection of that or it gets really irritated, it gets really red and inflamed, and that's known as conjunctivitis. Okay, that's all the cornea. Okay. And the cornea needs to be transparent because what do your eyes do? Well, they take light, right? They take light and they transduce it into an action potential, the neuron. So there's also transduction here via what are called photoreceptors. Okay, so you have your anterior chamber, you have this outer cornea, okay? And the anterior chamber is filled with a very clear, watery fluid, okay, and that is known as aqueous humor. And you guys have all had eye exams, right? You had that little exam where they put that little puff of air on your eye. Have you guys had that? It's called a tonometry. What they're doing is they're testing the pressure, something known as the intraocular pressure, and they're testing the pressure inside of this anterior chamber. There are diseases where you either make too much aqueous humor or you can't drain it off effectively. Okay, your intraocular pressure can get higher than normal, and it can cause damage. And common disease is something known as glaucoma. You guys ever heard of that? Glaucoma. Okay, and that is what's going on there, is that intraocular pressure is very high. So that's why they test that when you get your eyes examined. Okay, so it has to do with the anterior chamber. And then you have the colored part of your eye, what's known as the iris, right? And the iris is actually a band of muscle, okay? It's a band of sphincter muscles, okay? And they can contract, and they can dilate, or they can relax. They can contract and relax. And that causes what? Well, that causes the iris to close and open up, okay? And of course, the pupil, the black thing, the pupil is nothing but a hole in the iris, right? And when there's a lot of light, the iris constricts down and the pupil gets smaller to let less light into the eye. And when there's low light or less light, it relaxes, the pupil opens up, and that allows more light in. Does that kind of make sense? It also should be no surprise that when you have a sympathetic response, that fight or flight response, what happens to your pupil? It what? It dilates. Why? Why would you want your pupil to dilate? Huh? Yeah. Because remember, your sympathetic nervous system is that fight or flight response, right? If a bear, a lion or a tiger, a bear jumps out and it's like, hey, I'm going to eat you, and you're like, hey, I don't want you to eat me, right? Your pupils are going to dilate. You want your pupils to dilate because you want to be able to see everything that's going down to give you that advantage, right? So that would make sense that you'd have pupillary dilation, or something known as midriosis. You ever heard of that term, midriosis? That means to dilate, and then meiosis means to constrict. So midriatics are medicines that dilate your pupils, and meiotics are medicines that constrict your pupils. Okay, so you have that, you have your pupil here, and then what happens is as that light comes in, okay, that diffuse light comes in, that light then gets focused through the lens, right? You have a lens that focuses that light, okay, onto the back of the eye, okay, this structure known as the retina here, and we'll talk about a couple of the layers. And this can be a problem as well, right? As you get older, your lens can develop these opaque spots on it, known as cataracts. You guys ever heard of that, a cataract? A cataract is a problem with the lens, and of course, when you get these opaque spots, the lens is not able to focus that light, and then you get people like me and probably several people in the class, okay? And what can happen is your eye can get a little longer than normal, because as that light comes in, okay, that lens focuses that light. The focal point of that light should be right on the back of the retina, right? But if your eye gets a little long, what happens to the focal point of the light? It's not right on the back of the retina, right? It's in front of the retina. So the image is actually going to be very fuzzy. Does that kind of make sense? And that's something known as myopia. Myopia. You are myopic. Okay, that also a common term for that is something known as nearsightedness. Have you guys ever heard of that term? Nearsighted. That means if you're myopic or nearsighted, that means that you cannot see things very far away. You can only see things near you clearly. Does that make sense? And we use a scale, okay, to determine how myopic somebody is. And normal vision is 20/20. Have you guys ever heard of that term, 20/20? And all that means is that at 20 feet, if you have 20/20 vision, you would see what a normal person sees at 20 feet, right? Does that make sense? But if you're myopic, and let's say instead of 20/20, your vision is 2200, that means that what you see at 20 feet is what a normal person would see at 200 feet, right? Does that kind of make sense? And then you can actually have kind of the opposite of myopia. You can have, like say, 20/15, okay? So that means that at what somebody sees at 20 feet, you see it like it's at 15 feet, so your vision's a little better than normal. Does that kind of make sense what's going on there at that scale? It's kind of a weird scale there. Okay, cool. All right. You have three major layers. The outer layer of the eye So here we've gone through the anterior chamber. We're going to go through the posterior chamber, and the lens of the eye exists within this posterior chamber here. So anterior is up here, posterior is behind the pupil. You have the lens. The lens is attached to this muscle here called the ciliary body. Okay, see this muscle, ciliary body? It's not the iris. And it's attached by these little suspensory ligaments. And the ciliary body can actually contract and relax, and that can change the shape of the lens. And that allows you to focus, right? If something gets near you, that lens needs to change its shape to focus that light. And then as it gets far away, the lens will change its shape as well. And it's very quick how it can do it. Fractions of a second that what we call accommodation can occur. And then you move into the posterior cavity here. And the posterior cavity of the eye, behind the lens, is filled with something known as vitreous humor. And that's kind of a gelatinous, it's like a transparent jello. Okay, it has a very gelatinous-like appearance and feel to it, as you guys will see. All right, and sometimes parts of that can kind of break off or degrade, or you can get clumps of protein in that. And if you get that, like I have, little clumps of protein or blood cells in there, what happens is as the light comes through and hits those things, they'll cast a shadow on the retina, and that will create something known as a floater. Right? You get like a little dark, weird shaped thing floating around in your vision, and that's normally what would cause that. Okay, so the innermost layer here is what we call the retina, and the retina is what contains the back of the retina here. Okay, is what contains the receptors for the light. Okay, you have what are called photoreceptors. All right, and you have two general types of photoreceptors. You have rods and you have cones. The rods are not good at detecting color. Okay, the rods are your black and white and gray kind of vision. So your rods tend to be more peripheral, right? They tend to be more peripheral, and your rods tend to be better in low light conditions. So they don't provide a very crisp picture, okay? So they're not real good detail, but they're good for low light vision. They're more concentrated than the periphery. And the cones provide very crisp detail. And the thing about the cones is they can detect color, right? They are sensitive to red, green, and blue. And then different combinations of red, green, and blue, of course, can be used to create all of the colors within the visible spectrum. And remember, we talked about this at the beginning of the class. We can only see a tiny sliver, right? We can only see a tiny sliver of the electromagnetic spectrum. Basically, wavelengths of light from about 400 to 700 nanometers. Light that has a wavelength of less than about 400 nanometers, that goes into the ultraviolet range. And then when the wavelengths get even smaller, you're looking at X-rays, and the smallest and most energetic, of course, are going to be gamma rays. And then on the other end, wavelengths that are longer than 700 nanometers, you're looking at infrared. Longer than that, you're looking at microwaves, and then the longest wavelengths are radio waves. So most of the electromagnetic spectrum is outside of our ability to see, but we know that certain animals, right, such as what we call pit vipers, rattlesnakes belong to pit vipers, they actually have heat-sensing pits that can detect infrared light, whereas, of course, we can't. Okay, so the light basically hits those photoreceptors. Those photoreceptors send a signal to a bipolar neuron, and then the bipolar neuron sends that signal on to a ganglion neuron, and eventually it makes its way to the optic nerve. That signal then goes to the thalamus, and then the thalamus sends that information back into the occipital lobe, right, where you have your primary visual cortex. And then you have integration of that occurring there. Remember, you have decussation, So you do have crossover, right? Information from this eye crosses over to this side. Information in the left eye crosses over to this side. So you have decussation at the optic chiasma, which we talked about last week. Okay, cool. So just make sure that you're familiar. Remember, you have three major layers. Your outer layer is what we call the fibrous tunicula. Okay, and that contains the sclera, which is this part here, and then the sclera actually here is this part, and then of course you have the cornea here, okay, which is the anterior chamber, all right, and then you have this middle layer, which I draw in red here. Okay, that's your vascular tunic. That contains, of course, your blood supply to most of the eye. Okay, it contains the choroid, the iris, the ciliary bodies, all that. And then finally you have the innermost layer, what's known as a sensory tunic, and that's where your retina is. That's where you have what's also known as your macula, which is in the back of the retina there. Okay, you guys are okay with that? Some other problems you want to be vaguely aware of, something known as a retinal detachment. You guys ever heard of that? It's where the retina actually becomes detached, okay? And if it becomes detached, that means it loses its vascular supply, right? It becomes detached from the middle layer, loses its vascular supply. And if it isn't reattached, of course, the retina can then die, and you will have permanent vision loss as a result of that. You guys are familiar with something known as macular degeneration as well, where you have compromise of the blood flow through that area, and you end up losing a significant part of your vision, specifically the central part of your vision, is what we see with macular degeneration. So there are lots and lots and lots of disorders with the eyes. Looking externally, just a few things. Of course, you should be able to identify the eyebrow, which is good at keeping sweat and debris out of our eye. You have your eyelashes. Your eyelashes are very sensitive, right? You touch your eyelashes and that can elicit a blinking reflex to protect your eyes. You also have a lacrimal gland up here, and then you have a lacrimal sac, and that, of course, they secrete tears, right? And you have a constant, what we call lacrimation occurring, and that helps coat and protect the eyes as well. Okay, so that's that. Let's go ahead and let me show you the dissection of the eye. There should be one for each group. I'll just show you guys how to do that real quick. Point out a few structures. If you can get away with just using scissors, that would be great. And what I would recommend is, you see these really sharp scissors that look like this versus the kind of the blunt ones like this, I kind of recommend using these. If you have to use a scalpel, you have to, but you know, just be really careful if you're doing that. All right, and what I'll do is I'll turn the projector on. Okay, we'll project it onto the board. I think I've struggled with that thing the whole semester, haven't I? We'll get this thing going here, and we will turn the document viewer on. Give the old college try here. Hey, all right. Look at that. All right. Move this over there. Okay. Turn this on here. Okay, good. All right. This thing, let me get some lights going here. And we should be good to go. Lights. There we go. Alright. You guys see that okay? That's actually not too bad. Alright, let's see if I can zoom in on that eye a little bit better though. There we go. Okay, so here we have our eye. You guys good with that? So if we just take kind of a look at the external characteristics of the eye, you can see that I'm kind of moving. Oh, there we go. I'm kind of moving the anterior chamber around. So what's this outer part here? This is the cornea. Good. And then what this outer part here is the sclera, and you can actually see some of the muscles still attached. And then you can see the optic nerve right here. So what I would recommend you do is either make your cut or your incision, instead of kind of making a sagittal, we'll do a coronal incision. So what I want to do is I want to cut anterior from posterior so we can appreciate the anterior parts, the anterior chamber, posterior chamber, the lens and all that. So what I'd recommend is you just kind of go in. Oh no. There. How's that? All right. Let me I'll widen it out just a little bit. There we go. All right. So you kind of go just behind. All right, kind of toward the center of the eye. And see how I just kind of cut in? All right, so what layer am I cutting through right here? What's that outer layer? The sclera, right? So I'm just going to cut this, and I'm going to separate anterior from posterior. I'm just going to cut around here. All right. It says it's a lot safer using scissors as opposed to scalpel because this is such a small structure that we're dealing with. There we go. All right. So what's this blob of jelly here? Right here. So we're in the posterior cavity here, but what's this stuff called? What's this gelatinous stuff called? The vitreous humor, right? This is a vitreous humor. So again, this is the posterior part of the eye here. This is the anterior. So I'm going to pull that vitreous humor out. All right. And what do we have right here? Kind of this hard. It has a hard, and it fits right here, and you can see the pupil. So it's right behind the pupil. What is that? Say it again. Yeah. So we're looking at the lens there. All right. Good. And so we'll put the vitreous humor back in the posterior chamber. We'll identify some of the structures here. So what's this dark? Oh, there you go. What's this? Yeah, so this is the iris here. And then what's this muscle here that the lens is suspended on? What's that known as there? The choroid, right? It's known as a choroid there. All right, and then what am I looking at right here? What am I pointing to right here? What's that structure again? The cornea, right? So you've got the cornea here. Yeah, cornea, right? And you've got the iris, right? And you can actually see the iris a lot better now. So cornea here, and then iris is deep to that. And then you're going to have your lens right here, and you can see where it was attached by suspensory ligaments. Okay. And then you've got the posterior part. All right, so you've got this open here. You've got part of your choroid here as well. You've got this little structure here. Oh, You guys didn't tell me it was out of, okay. So you've got this little structure right here, okay, and this is actually the retina that you're looking at there, okay. And let's see here if we can really identify anything else. No, not a whole lot else. So those are the major structures that you'll just kind of want to be able to familiarize yourselves with, and there's a little picture in your lab manual as well that you can use to kind of walk yourselves through it. So let me go ahead and turn the lights back on, and if you guys want, grab a tray and an eye for each of your tables. Do your dissection, identify your structures, and then just kind of work your way through the lab, and I'll be available to help out or answer questions, anything like that. Like I said, I'll give you guys a little time to do that, and then we'll do a review for your upcoming exam.